Hi everyone, my name is Akash Sastry. The title of this talk is Integrating Coastal Zooplankton Monitoring Programs into an Essential Biodiversity Variable Framework, Current Status, Challenges, and New Developments for Canada's West Coast. My co-authors are Sonia Batten, Claire Ostel, and Brian Hunt. So the basic goals of this presentation are to provide an overview of the major zooplankton monitoring programs on Canada's Pacific Coast, with an emphasis on addressing how these data sets align with the EBV framework. So I'd like to dig into current status and some of the challenges and gaps. So a useful definition of essential biodiversity variables comes from Muller-Karger's paper in 2018, and they define EBVs as a small set of variables which collectively capture biodiversity change at multiple spatial scales and within time intervals that are of scientific and management interest. And here's a figure from that paper which nicely illustrates the relationships between EOVs, or essential ocean variables, and EBVs, and each of the 6 EBV classes. So I'll draw your attention to the EOV, zooplankton biomass and diversity, here in orange, and you can trace its relatedness to each of the various essential biodiversity variables on the right-hand side. So what are the EBV classes, and which ones are most related to zooplankton? Okay, so here's the table of EBV classes originally put forth by Pereira et al. In 2013. And these are the classes that were adopted by GEOBOM. So highlighted in green are the 4 EBV classes with the most connectivity to the zooplankton EOV, and these are the 4 classes I'll discuss in this presentation. So the first EBV class is species populations. This is a class describing variation of abundance and distribution targeted at the population level, so species of ecological interest. And it has a temporal sensitivity of 1 to greater than 10 years and demands relatively long time series. The second EBV class is community composition. So this is the class describing variation in measures of diversity such as taxonomic diversity and functional diversity. These indicators are sensitive to variation taking place on timescales approaching 5 to greater than 10 years and requires fine multi-species taxonomic resolution. The third EBV class is species traits, and the table here identifies a good example of a species trait, which is phenology, and which describes the timing of life history events for key species. So the temporal sensitivity of species traits is relatively high, at about a year. And finally, the fourth EBV class is genetic composition, describing allelic diversity for target species. So for practical purposes, we can distinguish between 2 types of zooplankton monitoring programs along the Pacific coast of Canada. This slide outlines the first type of monitoring program, which is ship-based sampling using vertical net hauls. The map identifies plankton stations within statistically defined regions. So the red symbols represent DFO stations, and these green symbols represent Hakai Institute stations. The longest time series is DFO sampling, DFO monitoring over the southern Vancouver Island shelf and shelf break, this SVI region. And this time series is now over 40 years old. Included in the SVI region are Line P coastal stations, which have been sampled in February, May, and August since at least 1990. The NVI, or Northern Vancouver Island region, includes DFO stations sampled seasonally for approximately 30 years, as well as Hakai Central Coast stations, which have been sampled since 2012. Further to the north is the Hecate Strait bioregion, which has been monitored on a seasonal basis for about 20 years now. And lastly, the Strait of Georgia time series is over 30 years old. However, it's a little gappy. And DFO monitoring here in red and Hakai monitoring has been taking place continuously on a monthly basis since at least 2015. So all zooplankton sampling identified here is effectively the same and consists of vertical net hauls of 236 micron plankton nets. Here's a picture of some bongos. And these are nets that are hauled from just above the bottom or down to a maximum of 250 meters to the surface. And the preserved contents of each sample are counted and identified down to species and often developmental stage. So the taxonomic resolution for this data set is very detailed. So the second zooplankton monitoring program is the North Pacific Continuous Plankton Recorder Program, which has sampled continuously in Canadian coastal waters since 2000. And here's a map showing the North Pacific CPR sampling in the region, which is also covered by DFO's programs. So it's worth noting that the overall North Pacific program samples throughout the North Pacific. The yellow symbols here represent samples collected in 2019, and the idea here is to give you an idea of the overlap with DFO monitoring. There are typically 6 to 9 surveys per year, which roughly correspond to monthly sampling between May and October. And pictured over here is the CPR tow body, which is towed at speed 5 to 10 meters below the surface by ships of opportunity. So animals are filtered through a 270 micron mesh and retained on silk. And pictured here is an example of an internal cassette housing that silk on rollers. There are about 7, for this survey, there are 6 to 7 silks per survey, and each silk is divided into sections corresponding to 18.5 kilometers or 10 nautical miles. The taxonomic resolution of this time series is also detailed, comparable to the DFO taxonomic resolution. So both types of zooplankton monitoring programs in the Canadian Pacific have key characteristics making them suitable for integration into an EBV framework. They are long-term time series. They consist of continuous monitoring of abundance and distribution, and all have detailed multi-species taxonomy. This is a cartoon from Kissling et al. 2018, and it represents the workflow from raw observations to EBVs to indicators. So generally the process starts by identifying the raw observations from different sources as EBV usable data sets, and then standardizing and harmonizing as needed for EBV ready status. So the question is, where are we in relationship to this workflow? Okay, starting with EBV classes 1 and 2, species populations and community composition. For species populations, which are typically target species, we need information on species abundance and distributions over contiguous spatial and temporal units. And both the DFO and CPR time series meet this requirement for key species indicative of ecosystem function, so they're termed EBV usable. For community composition, we need information on community abundance, taxonomic and phylogenetic diversity, and trait diversity. So again, both the net-based and CPR time series meet these requirements. They're both long-term time series with finely resolved taxonomic enumeration, and also termed EBV usable. Functional traits have been identified and diversity indices applied to both line P and the SVI time series. And since it's relatively new, I'm going to describe some of the work we've been doing on functional trait diversity over the next couple of slides. So functional diversity can be used to relate biodiversity and ecosystem function, since it characterizes the diversity of species traits rather than species identity. And these traits are phenotypic characteristics that describe or relate how organisms function. So ultimately, it's a tool used to explain variation in the structure and function of communities. I'm highlighting this area of the CBV class because several of my colleagues and I have been working on this topic for this region. So the first application of the functional trait approach for marine zooplankton time series was published by Pomerleau et al. In 2015. And this is a snippet of a table that represents our matrix of species and functional traits for the zooplankton community sampled in coastal Canadian waters. And we chose traits related to function, so adult body length, feeding type, and feeding types like active ambush feeding, cruise feeding, current feeding, trophic group, so whether an animal is a herbivore, an omnivore, or a carnivore, or somewhere in that continuum, reproductive mode. So examples of reproduction mode are broadcast spawning or egg brooding animals, vertical distribution, DVM, and habitat. So this categorization of traits was based on references in the literature. So this demanded a fairly extensive literature search. And this species trait matrix was then used to develop ecologically meaningful functional group classifications using cluster analysis, and this served as a form of validation. So the functional approach effectively renames species on the basis of their functional traits. We can use the species traits matrix I just showed you to calculate functional diversity indices, which are analogous to taxonomic diversity indices. This upper panel represents interannual variation of functional dispersion for coastal line peace stations sampled between 1997 and 2012. The functional dispersion is analogous to Shannon diversity and represents a multivariate dispersion of traits within a community. So here positive values represent an overdispersion of traits relative to the time series mean. The middle plot illustrates interannual patterns of functional evenness. And functional evenness is analogous to species evenness and represents the distribution of traits in trait space, where high values represent a regular distribution of traits. And lastly, functional richness is comparable to species richness. So rather than the number of species, this is the relative number of traits or the amount of niche space occupied within a community. So harmonization of the zooplankton time series. There are advantages to using both the DFO and the North Pacific CPR raw observations. However, we have some challenges to harmonizing the two data sets. And these are brought about primarily because of differences in the sampling instruments themselves, so the CPR and nets, and the depth at which they sample. And so just to remind you, the CPR samples the community primarily at the surface, and our nets sample down to seafloor, or at least down to 250 meters, which corresponds to the maximum depth of diel vertical migration. And so these differences are borne out in terms of abundance, presence, absence, and composition. And there may be additional differences in the data sets that are brought about because of differences in taxonomic resolution. So the condition of animals is not the same between the two programs, and some animals are not as easily identified given the way they were sampled. So there are some examples in literature of studies where they tried to reconcile CPR sampling to plankton net comparisons. So a direct comparison was made by Hunt and Hosey in the Southern Ocean. And this was a directed study where they compared depth stratified net samples to the CPR and addressed where the greatest similarity and greatest differences exist in sampling. And another study by Kane in 2009, which compared the CPR time series and the net-based time series in the Gulf of Maine. So if we were to approach harmonization for our region, we'd probably have to approach it in the context of both of these approaches. So species traits are the third EBV class, and this is effectively a monitoring of trait changes for key populations or target species through space and time. There's some overlap with the functional trait approach I described earlier, but it is different because this approach identifies traits which can and should vary over relatively short temporal and spatial scales. So there are 5 proposed species traits by Kissling et al: phenology, morphology, reproduction, physiology, and movement. And a good example of a target species is the large lipid-rich copepod Neocalyx plumcris. And this is a biomass dominant group in the Northeast subarctic Pacific. Its phenology is well studied because the populations develop synchronously and have clear stage structure, which makes it relatively easy to identify seasonal timing of peak biomass. And both the DFO and CPR time series have been used together for this purpose. And a good example is the study by Macus, Batten, and Trudell in 2007, and it provides a nice example of how phenology can be compared both in time and space using both data sets. Morphology can include body size measurements such as variation of prosome length or body volume, and this information already exists or can be mined from our archive samples. Reproduction measures the reproductive output, And this may be a little bit more difficult to approach, but might be modeled via early season cohort recruitment numbers for the species. Physiology, this might include any number of functional traits which I outlined earlier, but may also include fitness promoting traits such as lipid content. And finally, movement. So again, for Neocalus plumcris, the life history of this animal is characterized by a deep overwintering period. And the timing of descent and overwintering depth are potential traits which can be monitored through time. Okay, the last EBV class I will discuss is genetic composition. So these are measurements of genotypes or allelic diversity within key or target populations. And this is relatively new for our group, but we now have good zooplankton molecular capacity and expertise at DFO. And so we've been analyzing specimens from our routine collections and also from those shared by multiple organizations, which include NOAA and Oregon State University. So a good example is work done by Julian Smith and several members of our group in a paper that was published earlier this year, and it documented the abundance and distribution of Acacia tonsa, which is a warm water copepod on our coast. And this study took advantage of both detailed microscopic and molecular analyses using 16 sRNA sequencing for specimens from California, Oregon, and British Columbia to characterize the local genotype as being a mixture of southern populations. And detailed microscopic work confirmed this genotype as distinct. And a new species, Acacia bruta, has now been proposed. So this work is evolving, and we're now taking a similar approach with several target species. Okay, to summarize, so long-term zooplankton monitoring on Canada's Pacific coast meets criteria for EBV usable data sets. And I discussed 4 EBV classes most relevant to zooplankton biomass and diversity. The first 2 are species populations and community composition. And these consist of continuous measurements of abundance and distribution, temporal sensitivity, so time series greater than 10 years in duration, which is the case for most of our time series, fine taxonomic resolution, and functional trait assignments for the majority of species. So we have all of these in hand. And the next step is harmonization of these data sets, and I discuss some of the challenges associated with that harmonization. The third species, EBV class rather, is species traits, and I described phenology as well-studied trait for the biomass dominant group, Neocalus plumcris, as an example. And our next step would be identifying outstanding traits which can be reasonably assigned, either via direct measurement. So I discussed the idea of making regular measurements of body size or body volume for the morphology species trait, or else mining literature for these characterizations. And lastly, I discussed the EBV class genetic composition, which is emerging in our program, and we have developing molecular and taxonomic capacity, and we've started with early population level studies, and this work is ongoing. So thank you very much for taking the time to listen to this talk, and I look forward to the discussion next week.